Kátia Meneguzzi. Eu sou aluna de doutorado do programa de pós-graduação em fitotecnia da Universidade Federal do Rio Grande do Sul. E eu irei dar início à segunda e última sessão desta tarde, denominada de Melhoramento para Resistência a Estresses Bióticos e Abióticos. Atualmente, a agricultura enfrenta novos e enormes desafios, devido ao crescimento populacional, à conservação ambiental e às mudanças climáticas. Para lidar com esses novos obstáculos, os programas de melhoramento necessitam disponibilizar cultivares resistentes, tanto a estresses bióticos quanto abióticos. Desta maneira, os agricultores poderão ter uma maior produtividade devido a uma maior expressão do potencial produtivo da variedade, além de uma menor necessidade na utilização de produtos químicos. Para falar sobre este tópico, iniciaremos com a palestra do Dr. Danilo Lyra, da Hottis Lyra Research. Posteriormente, teremos a apresentação da aluna de mestrado Bianca Oliveira Machado, da Universidade Federal do Rio Grande do Sul, que irá compartilhar conosco um pouco do seu trabalho de pesquisa. E finalizaremos esta sessão com a palestra do Dr. James Holland, da North Carolina State University. Para darmos início à segunda sessão, iniciaremos com a palestra do Danilo Lyra, que é formado em Ciências Biológicas pela Universidade Estadual do Sudoeste da Bahia. Possui mestrado em Genética e Melhoramento de Plantas pela Universidade Federal de Lavras e doutorado em Agronomia pela Universidade de São Paulo. Danilo tem experiência com pesquisa em atividades clássicas de melhoramento e em abordagens modernas de genômica e estatística, onde se concentra na compreensão da arquitetura genética dos traços quantitativos. Desde 2018, é geneticista quantitativo da Hottis Lyra Research, onde possui extensas colaborações em trigo hexaploide. Agora, passo a palavra para o Danilo, que irá nos honrar com a palestra intitulada Dissecação genética de genes da via biosintética da trealose para caracteres relacionados ao rendimento de trigo. As questões podem ser enviadas pela seção Q&A de perguntas e respostas e estaremos atentos às questões para repassar ao palestrante ao final da apresentação. Danilo, você pode iniciar seu compartilhamento de tela e obrigada por ter aceitado participar do nosso simpósio.
All right, can you see that? All right, so
yes.
Thanks for the introduction, Katja. I will speak in English today, so it's been a pleasure for the invitation. I'm really happy to discuss and talk about what I have been working on these 2 years at Trehalose Research. So basically, I will stick with my presentation today talking about this genetic dissection of trehalose in wheat. It's a biosynthetic pathway related to abiotic stresses as drought and flooding. I'll discuss this later. Okay, so first of all, I want to mention a overview of some nice research institutes here in UK. As you can see, we have Scotland. I've just put here the laser. Yeah, we have in Scotland we have the Rosalind and we have the James Hunter Institute. And in England, we have 4 main institutes as John Innes, Earlham, and NIAB. All of them, they work with wheat, as wheat is the main crop here in UK, as probably you know that. So you will see a lot of my collaborations. I have a lot of collaborations with these research institutes as well. So they have a funding agents called BBSRC, the same as in Brazil, we have CNPq, CAP, so they have this as well. They fund a lot of the research, and they increased the funds this year as well because of the coronavirus. They're investing in sciencing. And the conference, they have a national conference called Monogram. It's more related to grains. It's a small to medium conference. It's also nice to attend this. And they also have lots of websites, nice websites. As you can see, we have the wheat training. Please check it out. This is nice. And if you want to buy some germplasm, if you want to buy some materials, just go to seed store. They have all of these materials available. Mapping population is also nice. So they have been working, studying with wheat, right? So recently has been published this science publication, the roadmap for wheat. It's an improved version of the reference genome. What does it mean? It means that we have the total number of genes, the positions, the length, and all of this available today. So we have around 100,000 K genes. As you can see, we have the 1.0. But the question is that we don't know most of these genes. We don't know their functions. We don't know their connections to the phenotypes, and if they are important or not to the agriculture crop breeding. So this is the wheat genome, as most of you probably seen this, just showing the most important genes found so far. We have the RHT genes in chromosome 4 as one of the green revolution genes, and we have the vernalization gene as well. So all of these genes are inside some specific pathways, and so we still have plenty of work to study. As you can see, we have only tiny variation selected for, and we have a big hidden variation inside that we still have to look for. And only by research we could explore this hidden variation. So in UK, they have this strategic program called Designing Future Wheat, DFW. It's a nice research. It's a coordinated UK breeding program. So lots of institutions, they work, they play together just to deliver a new germplasm and to develop breeding and markers toolkits, and also to have these RNA-seq co-expression data developed by John Innes Center. And Rothamsted is responsible for the phenotyping. I'll mention about this phenotyping platform. And all of them is combined, is linked with the plant breeding program, the private breeding companies. So as you can see here, so all of this DFW is inside the WISP. And what is the WISP? WISP is the national breeding program in UK. So possibly don't have this in Brazil. Each place they have their own breeding program, but here they have this more in an organized way. It's divided in 4 main pillars as development of land races, synthetics, and wide relative integration. University of Nottingham, they have a nice program with this, and development of elite lines. Of course, the breeding programs are most interested in this, and supported by the phenotyping and the genotyping. So if you want to check it out, it's also nice to go and check this website and click in each tab here, each circle, you see loads of information, nice information, and please go ahead and check it out. So as I said, Rothamsted is involved as well in all of these projects, and I've just mentioned very briefly about Rothamsted research. As you can see here is a nice picture of the entrance of the institute, and we have 4 main departments. We have the sustainable, I'm not mention all of them, but I'm allocating this computational and analytical science, the CAS, which develop all of the statistical works and also the bioinformatics stuff. But I also have loads of connections with the plant science, so in where they use more molecular biology on all of this stuff as well. And the institute, as I said, is most concentrated in the wheat. It's not breeding itself, more molecular biology in this kind, but they also have some works in camelina and oilseed rape. But well, the institute is well known, recognized. Possibly some of you might know that Ron Fisher developed his ANOVA model here in the institute. So they see Fisher as a guy that gave these large contributions to the scientific community, especially in stats. And Ron Fisher, he worked with this data here at the institute called Broadbook. It's one of the oldest agriculture field experiment in the world. So they also give lots of attention of this experiment. So inside Rothamsted, I have working as a quantitative geneticist. So basically I have many collaborations in different groups, as you have seen this picture. Collaborations with the microbiology group. We're trying to understand this relation with the rhizosphere and the microbiome. And the genomics group, how to use metabolomics data and multi-omics modeling as well. I will not mention about these two. Today, I'll just stick and talk very briefly about my collaboration with the phenomics group, and also a collaboration with Annie Phillips and Cristobal about the tealing population. And more specifically, I will talk about the trehalose genes. So I will split in three types, three kinds of presentation now. And the first of them, I will mention about the phenomics collaboration. Okay, so Rothamsted invested loads of money, more than £1 million in the development of this platform. Basically, it's a phenotyping platform, which is fully automated. It runs 24 hours per day, equipped with several sensors. So the main thing of this platform is the high resolution compared to drones or compared to other types of platforms, the high resolution it can give it to you. So this is a nice picture. As you can see, I took it from myself as well. So here's the area covered by this. Check it out, this publication, and also this website to know more about this. But the main question is, you might be asking is, why do we need this? Why do we need all of these investments? And the question is, why do we need phenomics itself? Roberto will talk about this more tomorrow. But the question is, we can use all of these high resolution cameras to monitor the performance and the plant health. More relates to biotic stress. I've not mentioned biotic stress. And it's a non-destructive analysis of the plant growth, morphology, and physiology. You can have equipped with lots of sensors to analyze the physiology as well. And also study key growth stages, again, with a spectral resolution. And most importantly, discover some QTLs or genes for dynamic phenotypes, which mean traits that are growing through time, okay? So that's why I'm going to show you some, the research that I have been developing, which is linking phenomics and genetics to discover QTLs. So basically, we use a mapping population in Chinese spring by Paragon and 26 time points, and for canopy height. We say canopy height because it's measured through cameras. So it's a 3D sensor, as you can see here. And what images it can give me. So basically, these are some images that you can see from the platform. You see here 3D images, nice images. And from these images, you can extract a measure plant height, measure the height. I didn't work with this data extraction of this. The phenotyping team with Malcolm and Nicholas and Pura, they did all the hard work. So I just used the 26 time points. So in a genetic perspective. So as you might know, we have high correlation, genetic correlation between time points in plant height. So basically I just modeled the genetic effects using a factor analytic model. And then the residual effects I just accounted for using the diagonals, the variance covariance matrix. And we used a B-splines to smooth, to reduce the noise between time points. And as expected, the variance and covariance increases over time here. And our main question of this publication was to run functional QTL mapping and single interval mapping. So as you can see here in the picture, you have individual time points, composite interval mapping, and we detect some transient QTLs. QTLs detected in only one time point, as you can see the 2A. QTLs detecting in persistent QTLs, we call this because it's detected throughout the crop development. Okay, so we have here the functional QTL methods. As you can see, the chromosome 5B, it was detected, was an additional QTL detected only by functional QTL models. So we wanted to see and simulate some data. Why do we have to use these types of approach? So yes, I just simulated the data used for persistent QTLs or QTLs detected throughout the time. So basically you have slightly advantage, slightly statistical power when you use these models compared to individual time points. But for the transient QTLs, you have a higher statistical power when you use individual time point, interval map in this case we used. So basically you can use both types of approaches, it's easy. But I also run some different strategies for genome prediction. If you want to check it out, this publication, please go ahead, it's published in JXB. Okay, so I will talk about my second project, which is related to the tilling population, in collaboration with the John Ennis, Cristobal, and Phillips. They were responsible for creating all of these lines. And we wanted to estimate the contribution of ultra-rare variants in this panel. So what's the tilling population? Tilling population consists of lines, mutagenized lines. So each line has hundreds of thousands of mutations, right? So basically, if you take all of these mutations and you want to understand all of these mutations, researchers have been using this to knock out some genes and to study their functions. So this is a nice way. It's not GMO, so be cool. And we have around 100,000 K genes reference using the new reference genome. We have around 4 million mutations in total. And all of these mutations are inside ensemble plants, okay? And all of them are, you can see these types of consequences. They were annotated. So basically you can see all of these annotations and non-coding, synonymous, non-synonymous inside the ensemble. If you want to just type the ensemble plants, and you will see the same population there. So our main question here was to develop a quantitative genetics pipeline. How we can deal with this population in a quantitative genetics perspective. So basically we want to answer some questions like, what's the association? Can we detect some genes with the combined mutations in the gene? Study pathways, what's the heritability of the most target pathways, and study, of course, the networks for the biological knowledge. And if you have the main question, the most important question I'm asking here is to study the gene dynamics through time. If you see, I studied the QTL, but QTL is a region. We don't really know. We'd have to do fine mapping later to discover the genes and target them. But the question here, we are working with the gene itself. So to answer this, we had this population in the field. Last year, we have around 800 mutations, 800 lines, and we are going to answer this. I already have some results. We partition in non-synonymous and synonymous mutations. As you can see, we detect some genes using only non-synonymous and using only synonymous mutations. I did not detect using non-coding. And another nice result is that you can estimate the variance explained by that gene. And when I combine the total number of mutations, we have genes with 800 mutations explaining basically nothing of the variance of plant height. This is for plant height. And I also partitioned the variance by chromosomes. If you see, variances explained by the chromosome is increased when you have more mutations, and also, of course, when you have more the chromosome length. Okay. So basically, there is a project that I'm still working on it. So it's a nice project to see this contribution of ultra-rare variants. And of course, please check it out later in the future. It might take a while to finish all of this. Okay, I gave you loads of information. Possibly you are just ruminating all of this information now. But I want to mention this genetic dissection of the trehalose genes, okay, which is the most important for us because it's related to the abiotic stresses, okay? So what is a genetic dissection? First of all, we have to access this genetic architecture. When you see the genetic architecture, you finding associations between genes and traits. So it's a way to dissect the trait and estimate the contribution of that gene and estimate and ask if the gene is under selection or not. And can we use all the gene effects to predict the traits? As prediction, we have seen most of the presentations today. So we're just using, I'll show our strategies as well. So I just want to mention that this project was in collaboration between Rothamsted Research. Matthew Poole, he's in charge of all of this understanding in biology of the trehalose, the Earlham Institute, and the CIMIT, Gamma Moledo, and Matthew Reynolds. Okay, I first want you to understand what's a biosynthetic pathway. It's very easy. It's just a network of interacting genes. Genes are interacting between each other all the time. We all know that. But the main key message here is just we have a bunch of molecules and enzymes just working with each other to produce a final molecule. If you take this final molecule, but I'll show in our case is the trehalose. So the trehalose in plants, if you see, they have in plants, they have also in animals. It's a it's a pathway related to the carboallocation. So if you think about the starch, carbohydrates, all this translocation is related to the trehalose genes. You also have the SUC biosynthetic family. So it's another strategy I'll just mention about the trehalose. Okay. So I will not explain the whole mechanism because it would take a while. So I just wanted to keep in mind that we have TPS genes and we have some TPS1, 6, some classes most closely related to Arabidopsis. Okay. So after the TPS, they produce a T6P molecule, which will activate the TPP genes, which will eventually produce the trehalose. Okay. So these trehalose genes, they are associated with a drought stress. So Kara Griffiths, she with Matthew Pohl, they developed this chemical intervention of T6P. So basically you take this T6P, they just isolate this chemical and they spray in the plants in the field. So when what they observe is that you have a nice recovery when you have vegetative growth after drought. And when you have well-watered conditions, you can see as well some increase in grain size because of this trehalose and carbohydrate allocation. And they also, this is not Matthew Pohl's group, but they also published in maize a overexpression of a TPP gene related to the recovery as well to severe drought. So again, keep in mind that is fairly related to abiotic stresses. So we take all of this information from the trehalose and we just sequence all of the genes using a sequencing technology called Exome-capture. The Exome-capture is not only exons, okay, we also have a different terminology for that, but also accounts for the introns. So that's what happened in our case. We have more non-coding substitutions. For instance, we have more introns inside the genes than missense mutations. So it's a trick, but I will not show here, but we have more associations related to the introns. So basically non-coding substitutions are playing a high contribution to the phenotypic variance. But before any analysis, you just have the genes, the list of genes, you just send this to the sequencing. But before that, you can have a look at your genes and their connections. You can use this knowledge network. It's a NetMiner, a software developed by the bioinformatics group at Rothamsted. If you want to know, check it out. This website is nice. And I just run the TPS11. You have 3 genes, the homologues, right? And then you have nice interactions between other traits and genes. So you can measure what's going on with these traits, what their interaction. So I combine this marker data with the phenotypic data. The phenotypic data, we used a biomass mapping population from cement, developed from cement, consisting of elite and exotic materials. It's published by Molero. As you can see, we have some nice differentiation as expected using the whole genome, the 35 Ks in shape, and using the exome trehalose. And I will mention about these results right now. So just to refresh your mind, we wanted to dissect the traits. And one of the main questions was to identify the connections between the trait and between the phenotype, the gene. So for this, you can use the traditional way, just like a GWAS, you ask if the SNP has an association with the trait. But in our case, we have the gene, number of variants inside the gene. So in this case, use an approach called gene-based scanning. It's well known, used in human data. As you can see, it's a variance component model, a scat and scatter. It's well known in models. As you can see, we have the genes at the TPS, TP6, and TP7, all correlated with the phenotype. And as you can see, we have some correlations for plant height using the TPS1. And also you can see some evidence of pleiotropy. What is pleiotropy? It's one gene basically controlling different types of phenotypes, right? Because they're basically correlated, okay? So the other question, we want to see the upstages. Okay, it's a biosynthetic pathway. They should be interacting with each other. That's more reasonable. But the question is that we detect some nice associations between the TPS1 and with some TPP. Basically, if you want to do some gene editing in the future or kind of this, you can target these genes possibly. And basically all of these interactions were positive interactions, meaning that you enhance the plate because you have the negative epistasis as well. These want to increase the plane height. And we have another question, which is what's the contribution of this gene to the phenotypic variance? So I used this model, GRAMA, with gene heritability, and we came with these strategies, also published in the literature. When you combine your GRM, or your genetic relationship matrix, coming from the genes, in combination with the whole genome, okay? Because quantitative traits, they are controlled by several traits with minor effects, some with major contributions. So you're just think, what's the contribution of this gene when I have all of these genes, okay? So we took biomass, for instance, here's just density of the beta distributions. And if you take the variance explained by the genes, you hear the number of the genes, the variance per gene, and we detect the 1D chromosome, 1D gene for the biomass. And what's the variance explained by this gene? It's around 12% of this gene. But we all know that we have the homologous, which means you have the same gene in 3 chromosomes different chromosomes. That's the main issue with the wheat. It's an hexaplod. You have this redundance. So this is the question. So we just observed that these trehalose pathways is contributing to the phenotypic variance of the biomass in this case. I did for all of the traits. So just taking the link between one point with another, we also run each SNP, asking if each SNP has an association with the trait. So we used the single variant analysis. So basically we found 2 missense mutations. Missense mutations in chromosome D. As you can see, you have the gene in exon 6. So basically it's changing the amino acid and something is happening because they have some effect on the trait. So that's why we wanted to see this observation between the trait, and we just classify this in analytic combinations. So we identified only 3 lines carrying these missense mutations. So we observed that consistently increase the plane height, the peduncle length, but decrease the grains per meter square. So as expected, because they are negatively correlated, right? So we are particularly very, very interested in the TPS1 gene. We were expecting to have at least 1 increase in the grains because methylpole is highly interested in the grains, but that's the result that we have. So we also asked if the genes are under selection because we have this breeding population, and the breeding population means that something is going on with the selection. Some genes are being selected for and so on. As you take a population, even a natural population, you have this kind of forces trying to change the allele frequency of the genes, and some genes are important to the phenotype, okay? So if you take the selection itself, it leaves traces in the genome. This is very well known. We have loads of publications. And if you have beneficial alleles, missense mutations, okay, you can call this as positive selection. But most of the mutations that happens is deleterious. So basically you see loads of negative selection, which means that you're just discarding, getting rid of all of these mutations, okay? So basically I just use this approach. You're just measuring the strength and the mode of selection in these genes. And we use the traditional approach called non-synonymous per synonymous substitution, very common. We use these RPECs. And as you can see, the TPS genes are mostly under negative selection. And basically that most of these mutations happen in the discarding or eliminating all of these mutations. This is okay, it's natural to happen this. And some TPP genes are related to the positive selection, which means most of these mutations they were selected for. Especially some genes are connected to some phenotypes. So this is also nice. Mostly saying that these trehalose genes, they're playing a huge contribution or even to the phenotypic variance. So my last slide, basically, we also wanted to ask if using trehalose genes to predict the traits, is it important or no? What happened with this? Traditionally, we use the SNPs in the whole genome, as I said. You can use CNV as well. I use this transcriptome and metabolome. The researchers are using this, but they are kind of expensive, and the values of the gains of predictive ability, they're not good, the investment of the money. And you have a nice strategy. When, like, we call this gene-based prediction, there is a nice publication, gene-based breeding, published this year. And this is the same way as I did for the gene heritability. Just you combine the gene with the whole genome, because you account for both, okay? So we identified that when you account for the 35 plus the trehalose genes, you have a slightly gain, as you can see here, for grains per spike, slightly gains for the grains filling, and a significant contribution for grain weight. As I said, it's related to the sugar content. So basically, if you take the sugar content, the grain weight, despite not having detected this in our association, it showed here that if you put this information in the predictive model, you could gain at least some predictive ability in this, okay? So my take-home message is that trehalose genes regulates carbon use and allocation needs are targeted to improve crop yields. We also have some future studies, in this case, to spray the T6P, what happens with the gene expression, what happens with the gene variance and all of this. And when we combine it with the drought stress, we have been working on this. The team have been working on these conditions, and we possibly are going to work in the future. And what's the gene by environment interactions? You mentioned a lot about the genotype by environment interaction. You want to see what's this connection with these genes specifically to the environment. And also the interaction with other pathways, okay? If you have several tons of pathways, so basically what's the interaction between trehalose and, let's say, auxins, trehalose, auxins pathway, and so on. The second is understanding the function of wheat. Genes pathways will accelerate crop improvement. As I said, you have some strategies, nice strategies, and integrating genomic and phenomics data will unlock the hidden variation of wheat that I said. And why not studying pathways during the crop development and so on. I would like to thank my network, especially Ganch Lavov, who is now in Sion, New Zealand, and all of these my collaborators that I have. Thanks for your attention.
Thanks for your presentation, Danilo. Obrigado pela sua apresentação.
Obrigado.
Nós teremos agora